Okay, today we're going to talk about DNA replication. So how we actually make those copies of DNA to put into our new cells before our cells can divide. Okay. So DNA has to be copied before a cell can divide. If the cell divides and the DNA has not been replicated, it's going to result in a cell that's not viable or a cell that has many, many mutations. And it is actually copied during a phase of interphase that is called the S phase, which is the synthesis phase, and we'll get more into that later. And new cells need identical DNA strands. So each one of your cells holds all of your DNA. Each one of your somatic cells holds all of your DNA, and that DNA should be identical to all of the other cells. DNA replication begins in an area called the origins of replication. And there are two strands where the two DNA strands are pulled apart that creates a fork, and that is called the replication fork area. At those forks is where you get the new strands of DNA that are laid down. So looking at the DNA strand that has been pulled apart at these hydrogen bonds, which would have originally connected these, you have this area called the replication fork, and this is going to be where the new strands are laid down. Okay? Dante. As the two strands open, they create what are called replication bubbles. And if you're looking at a prokaryotic cell's replication, they only have one single bubble. They have less DNA, and that DNA gets replicated much faster, but it also leaves more room for errors, which is why you can see evolution occur faster in prokaryotes. Eukaryotic chromosomes have more DNA, replication is slower and quite a bit more careful, and they have many bubbles. So at the bottom of this slide, these would be the bubbles. So you have these replication forks, and these bubbles are created around these replication fork areas. Okay. In order to separate these strands of DNA at those hydrogen bonds, you need an enzyme that is called helicase. That helicase is going to separate that helix, that helical structure, and it breaks those hydrogen bonds. Once those strands have been pulled apart or those hydrogen bonds have been broken, you need something there to prevent them from hooking back together, because those nitrogen bases are attracted to each other chemically, and you have this protein called single-stranded binding proteins, and those are going to help keep those unconnected and prevent them from going back together. You also have another enzyme called topoisomerase, which attaches the two forks of the bubble, and it helps relieve stress on the DNA molecule as it separates. This is showing the enzyme here helping to relieve stress on that forked area. Before you have a new DNA strand that can form, there needs to be something called RNA primers. And these RNA primers help start the addition of these new nucleotides. If you recall from the last lecture, a nucleotide is the subunit of DNA, and it's a sugar, a phosphate, and a nitrogen base. You have an enzyme called primase that actually makes that RNA primer. So primase makes a primer. When we're talking about these enzymes, pay attention to the ASE. Remember, enzymes have ACE at the end many times. And then you have an enzyme called DNA polymerase, and this is going to help add those new nucleotides as we create these DNA polymers, which are these long chains of nucleotides. Okay. DNA polymerase works in a way that it can only add nucleotides from a 3' end of the DNA. So remember, the strands run in opposite directions from each other. So here you would have a 5' end and a 3' end. You can only have DNA polymerase can only add things to the 3' end, which in this case of this strand here would be right here. So that's going to create a problem for the other strand. So new strands are built in 5 to 3'. Recall from our last lecture that we have those carbons that are numbered, and you have your 1, 2, 3, 4, 5 carbons. This is our 3', and this is our 5'. And DNA runs in that anti-parallel structure. Here you're seeing the 5' carbon and the 3' carbon. And here's our 3' here, with the 5' being on the bottom. You have two different strands. One of the strands is referred to as the leading strand because new DNA can be copied continuously, and it's going to go from the point of origin and towards the opening of the replication fork, or that Y that is formed in DNA when it's pulled apart. So it's going to go from the point of origin towards the replication fork, which is right here. And we're going from this 5' to 3' because DNA polymerase can only add at the 3' end. The second strand is called a lagging strand, and this is not a continuous replication because it has to copy this DNA in many short segments because polymerase can only add to the 3' end. So it's going to go from the replication fork towards the origin, which in essence this DNA has to be copied backwards. So this is our leading strand here, and it's going from 5' to 3' with the DNA polymerase adding it at the 3' end. Here we have this strand running in running in a different direction, and so it's having to be copied backwards. On the lagging strand, since it's copied in small fragments, these fragments are called Okazaki fragments. And then those Okazaki fragments must be joined together by an enzyme. Okay. So here would be this Okazaki fragment on the lagging strand. Here's the RNA primer. And then you have DNA polymerase, which is joining these together. So these little fragments right here with RNA primer, must be connected together. They're connected together by an enzyme called ligase. And this is going to form to make one strand. So here would be this ligase right here. So here's an Okazaki fragment, and here's an Okazaki fragment on this lagging strand. Okay, you see here, the leading strand is going from the point of origin, from this 5' to 3' end to the replication fork. And our lagging strand is going to go in the opposite direction. So it's going to go from this 3' almost to 5', and you have to have these ligase to hold these Okazaki fragments together. Okay, that new DNA must be proofread. DNA polymerase makes a mistake about 1 in every 10,000 base pair errors, which is pretty common if you think of how many base pairs we have in our DNAs. However, we have an enzyme that actually goes through and proofreads and can correct these mistakes before the DNA and the new cells are made. With the enzyme that helps proofread it, the error decreases to about 1 in 1 billion, which is much better. The idea of replication was presented by Watson and Crick after they created their model of DNA. The idea that the strands could be unwound and broken apart at those nitrogen bases. And how this is going to work after they've been copied, you have one original strand and you have one new complementary strand. So as the DNA is replicated, it's going to lay down a complementary strand for each of the original strands. So you have one parental strand and one new strand. So if you had your parental DNA right here, you would then have your new DNA here. So your original strand and your complementary new strand. So all of your DNA should have an old strand and a new strand. DNA can be damaged by chemicals and UV radiation. And you've seen that with UV radiation with things like skin cancer. Damage to those cells does damage the DNA, as well as chemicals. When people are exposed to chemicals like asbestos, as well as people who smoke with the chemicals they inhale into their lungs, that can cause lung cancer. And changing of our DNA can cause these cancers to occur. The cells must continuously be repairing damaged DNA. The more stress you put on those with outside chemicals, the more harder your cells have to work. There's a thing called excision repair, and it can go through and remove 50 damaged parts of DNA. And then you must use DNA polymerase again and DNA ligase to replace and bond those new nucleotides together. Okay, I'd like you to answer this in your notes, please. What would be the complementary DNA strand for the following DNA sequence if you have CGTATG? And use your base pair rules to do that.